Hello students, welcome to this session, the practical Zeal-Nielsen staining. Zeal-Nielsen staining is done for detection of acid-fast bacilli. Examples: Mycobacterium tuberculosis, Mycobacterium leprae, Nocardia. The reagents used in Zn staining, the first one is carbol-fuchsin, which is principal stain. The 25% H2SO4, which is decolorizer, and methylene blue, which is used as a counter stain. The procedure of Zn staining: so first prepare smear from the sputum sample, then start the staining. So first flood the smear with the carbol-fuchsin, and then you have to wait for 5 minutes, and in between gently heat the smear until the steam is seen, and then wait for 5 minutes, then wash. The precaution taken in the principal stain carbol-fuchsin: do not let it dry, and then whenever necessary more stain is added, and then when you see vapors, then you stop heating and then continue the primary stain. The role of heat is here the mordant, which allows more penetration of carbol-fuchsin into the cell wall of the acid-fast bacilli. Then wash the stain with running water. Then second step is decolorization, that is done with 20 to 25% sulfuric acid for 1 minute. Then next step is counter staining. So cover the smear with the methylene blue for 1 minute, then wash, dry, and then put oil and see under oil immersion lens of the microscope. After the Zn staining is complete, the result or observation: the acid-fast organisms are seen bright red in color. Because of the acid-fast means they are resistant to the acid treatment, so the carbol-fuchsin color remains, which is pink or red in color, and the counter stain is methylene blue, so background is the blue in color. And non-acid-fast organisms, like other than mycobacteria, staph, strepto, E. coli, klebsiella, or other organisms or other cells like lymphocytes, neutrophils, or epithelial cells, they appear blue in color. The RNTCP grading of sputum sample is done. So negative when no acid-fast bacilli seen, scanty when 1 to 10 acid-fast bacilli seen in 100 oil immersion field. 1 plus is 10 to 100 bacilli per 100 oil immersion field. 2 plus is 1 to 10 bacilli per oil immersion field, and 3 plus is more than 10 bacilli per oil immersion field. The clinical significance of grading is helping to monitor the prognosis of patient on the anti-tuberculosis treatment. The principle of Zn staining: the organisms like Mycobacterium tuberculosis, which causes tuberculosis, and Mycobacterium leprae, which causes leprosy, are acid fast. That means they are resistant to acid treatment because of the cell wall composition, which contains lipid like mycolic acid and few cholesterol esters. So they bind with the carbol-fuchsin tightly and resist decolorization by acid, that is H2SO4 used. So the pink color of the carbol-fuchsin remains, and they appear pink or red in color against the blue background of the counter stain, that is methylene blue. So sample for the diagnosis of pulmonary tuberculosis is sputum, and according to RNTCP, that is Revised National Tuberculosis Control Program, 2 sputum samples are collected, 1 spot and 1 early morning. So early morning sputum sample gives maximum concentration of the sputum, so early morning sample is preferred. The concentration technique used for the sputum sample is Petrov's method by use of 4% sodium hydroxide. The modifications of Zn staining: one is cold method, that is Kinyon's cold acid fast staining. In that, increased concentration of carbol-fuchsin is used so that heat is not required. Then second is different concentrations of H2SO4, like 25% used in Mycobacterium tuberculosis, 5% used for the Mycobacterium leprae, 1% used for the Nocardia. The other microscopic methods are fluorescent staining, that is use of oramin rhodamin dyes. Then other methods, one is culture, so it can be conventional method by use of use of the Lowenstein-Jensen media, but disadvantage is that it takes around 6 weeks time. Then the faster method is automated method, in that we can get report within 2 to 3 weeks. And the molecular methods used are PCR, Gene Expert, TrueNAT. So in that you can get reports within 2 to 3 hours. The tests to detect latent tuberculosis are Montauk's test, which is seen after the 48 hours of the given injection of the tuberculin. And then second test is gamma interferon. These are my references from we can study. I hope you find this session useful. Speaker Dr. Rajashree Mutha. Thank you.